Thank you, Jeremy. Welcome to everyone. I'm very pleased to be able to present this, since this is one of my major interests and has been for a long time. So the plan of this talk is to give some of the basic biology and microbiology really of microsporidiosis, and then and not spend too long on that, and then present some clinical cases, and along the way show how we identify the organisms and make the diagnosis. Yes. So I think I will, if you don't mind, stop the video because I feel very self-conscious with myself watching me. Okay, so let's go. So there's a lot on the slide, but I assure you I'm not going to read all the small print, but some of the basic taxonomy and phylogenetics of Microsporidia. These are obligate intracellular parasites, and they are now fully accepted as having evolved within the kingdom of fungi, but at a basal branch of that tree. In other words, not mainstream fungi. And if you look at the tree of life of eukaryotic organisms, at this orange arrow here, you can see where Microsporidia fit in, depending on which author you read, either a member of the Cryptomycota or very closely allied to the Cryptomycota down here at the bottom of the fungal tree. So these are very interesting parasites in that they have a remarkably reduced genome. In fact, the smallest eukaryotic genome ever described. The smallest one of this group is only 2.3 megabases, I think, which is really very, very small. They've got reduced mitochondria. They've got very small ribosomes, similar to bacterial ribosomes, actually, and various organelles like the Golgi body and so on have been simplified through evolution. So they were first identified 160 years ago, 1865, as the cause of a disease of silkworms called pébrine, which means pepper disease in French. And if you look at the bottom of on the bottom right here, you can see a healthy silkworm and a silkworm with a disease. And the serious looking guy next to them is Louis Pasteur, who was tasked with controlling this disease because it was a major economic issue for the silk industry in France at the time. And he solved the problem largely through implementing basic hygiene regulations and procedures in silkworm farms. Now, these organisms are well known to cause economically important diseases in bees, in farmed salmon, other farmed aquatic animals like shrimp, farm animals, and companion animals. And they emerged as opportunistic pathogens around about the middle of the 80s in the AIDS era. And from a medical point of view, it's predominantly immune deficient individuals who are at risk, but immunocompetents can also be affected. So there's an enormously wide host range for these parasites, from very simple protozoan organisms all the way up to high vertebrates. And some species are very highly specific in their host. For example, there's one species which will only parasitize one type of soil nematode. And there are many, many thousands of different kinds of soil nematodes, but some are more general pathogens. And generally, those are the ones that that we find interesting from a medical point of view. So this is a very large phylum. Currently, about 1,400 species are recognized in nearly 200 genera. Now, out of this enormous pool of pathogens, of organisms, 17 are currently recognized as causing disease in humans. And of those, some are much more common than others. So these are most of the important currently recognized pathogens. And without going through all the names in this list, I have put a box around the important ones. Encephalitozoon cuniculi, Encephalitozoon helem, the E. intestinalis, and Enterocytozoon benusi. But if you look at the central column here, you can see the wide range of organs in the body that can be affected. So ranging from eye, skin, muscle, brain, urinary tract, respiratory tract, etc., etc. And you'll see that the ones I've outlined as important, in fact, have quite a wide range of disease potential. Really, the only ones missing in this group, the only organ missing in this group is skeletal muscle. Otherwise, pretty much they can infect in one way or another just about every organ and tissue in the body. But they do have a propensity for certain systems, depending on which species we're looking at. And just to give you an overview of, well, firstly, this gives you an idea of the wide geographic range of these organisms, and basically they are wherever you look for them. And this was a meta-analysis looking at published articles, and the heat map shows areas of higher and lower prevalence as reported in these articles. So this doesn't necessarily reflect the actual geographic prevalence. It's simply the prevalence within the studies that were published from those countries. Now, interestingly, if you look at South Africa, you'll see that there are quite a lot of articles, quite a number of cases described in articles from South Africa, 167 actually, which is quite surprising. In fact, the highest number of any country on the African continent. And if you look on the left here, you can see those are the common pathogens which I've outlined previously, and at the top, the sort of hosts that have been investigated. So mammals of various sorts, bird, water, and we mustn't forget, although it's not shown on this graphic, insects and fish are also important hosts of these parasites. So how do they work? So the unit of infection for Microsporidiosis is the spore. And starting at the top left here, you can see the components of the spore, and the really obvious part that stands out is this coiled up filament or tubule inside the spore. And then there's a nucleus of course, because these are eukaryotic organisms, and various organelles and structures which have evolved to optimize the infectivity of this structure. The next picture along is just a 3D model of what a spore might look like, with this coiled up polar filament inside. And the polar filament is the route by which the microsporidial spores get into a host cell. So if you look at the central panel, the microsporidium shoots out its polar tubule, and the various triggers for this, including a change in pH and a change in permeability of the spore. So the contents of the spore, the pressure inside the spore increases, and it ejects the polar filament. And this is analogous to a rubber glove with the fingers inside and being ejected by air pressure. So if you look at the little video on the, will this play? Gosh, I hope this plays. I think I must do this. Sorry. If you look at the little video here, this is a demonstration of how that works. A rubber glove with the fingers inside increasing in pressure, and the tubule shoots out like that. So that's exactly the same process. Well, that's analogous to the process that that naturally happens. So once the polar tubule has penetrated the cell, the nucleus is injected and starts multiplying to land up with a collection of spores inside a vacuole inside the host cell. An alternative route is if a phagocytic host cell engulfs the spore, so therefore it's sitting inside the cytoplasm in a vacuole, and it then can, going through the same process, inject its germ plasm into the cytoplasm, and the end result again is the same. On the top right here, you can see an ejected polar filament from a spore in a microscopic preparation which has been stained, and at the bottom right, the same thing shown under transmission electron microscopy. So the spore over there on the bottom right hand corner, and the polar filament or polar tubule has been ejected and penetrating a host cell. So once inside the host cell, so here we are, this is the process we've been talking about, with the sporoplasm, basically the nuclear material inside the host cell. Then there's a process of division, cell division, going through a meront and then sporon stage. I think that the details here aren't important, but the mature spores land up inside these vacuoles inside the host cell, and they are then available to be released and then proceed with their job of infecting new host cells. So when did we first start finding Microsporidia in this country? We published the first case from South Africa in 1998. This was a patient at Sizwe Hospital. So some of you might remember Stella Pendle and Leonard Sachs. They worked at Cesbury Hospital when it was still an infectious disease hospital rather than a dedicated TB facility. This was done on basic morphological microscopic identification of the organisms, and I'll come on to show you how we demonstrate these organisms. Then 2 years later, we published the first electron microscopy micrographs of an organism, which really is the definitive microscopic method of demonstrating the microsporidial organisms, and allows you to at least make some shot at identification when you can view the internal morphology. So this was really in the days before widespread use of molecular methods. The first author on the EM case was Anne von Gottberg in 2000. So this first patients had diarrhea, and microsporidial diarrhea is really the most common form of microsporidial infection. And there are two species which are mainly involved, particularly Enterocytozoon benusi, and to a lesser extent, Encephalitozoon intestinalis. Now, E. benusi is a very ubiquitous pathogen. There are more than 200 genotypes across humans and animals, and it's very likely that some of these are zoonotic. In other words, animal pathogens which humans can acquire. They're not dedicated to intestinal infection, because both species can cause extraintestinal infection. Now, immunocompetent individuals can get microsporidial diarrhea, and it can be quite a prolonged illness, up to a month or even 6 weeks, but it is self-limiting. So in this regard, it's somewhat like cryptosporidial diarrhea, which can certainly not confined to immunodeficient individuals. And these organisms are a recognized cause of traveler's diarrhea. Now, if you look at asymptomatic carriage, in some populations, in fact, it's remarkably high. And a study from Cameroon showed that 68% of healthy volunteers in that country had carried microsporidia. But leaving aside the immunocompetence, the main risk group for microsporidial diarrhea are the HIV positives and other immunocompromised people, those with the usual organ transplant malignancies, hematological malignancies, chemotherapy patients, etc. Now, if you look at the global pooled prevalence of microsporidia in HIV-infected persons, it's been estimated as 12%, and somewhat higher in sub-Saharan Africa at more than 15%. If you stratify prevalence by income, low-income countries tend to have higher prevalences, and higher and middle-income countries somewhat lower prevalences. Sources of infection of these organisms, it's very likely that food and particularly water play an important role, particularly where sanitation and hygiene are poor. hence the association with lower income countries and to some extent with travelers' diarrhea. Where do these contaminants come from? Well, they come from infected hosts like insects, poultry, livestock, pets, and various food animals. Right, now, how do we identify these organisms in stool? Well, it's not that easy, but there are some things we can do. They are very small and tend to be overshadowed by all the other organisms and things that you find, debris that you find in stool samples. But we can, to some extent, show them up by using various stains and dyes. And starting at the top left, you can see these bright electric blue structures. Now, these are microsporidial spores which have been stained by calcifluor white, which is classed as a fluorescence whitener. And this binds to chitin in the spore coat. And with the right wavelength of UV light, it gives this bright, very bright fluorescence. Now, this is quite a clean prep. Usually, there are many other things in stool which also shine up brightly, including small yeasts. And so it's not terribly useful as a screening method. In the middle picture at the top, you can see the arrow pointing to what looks like something fairly nondescript, but that in fact is a microsporidial spore stained with a modified trichrome stain. And at higher power, you can see some internal structure which tells us that this is a spore and not something else. At the top right, another type of stain called a chromatrope type stain, and which stains at a slightly different color. And this is at a higher power. And in fact, if you look carefully, you can see a little pale area within the spore. And this is a vacuole, which is an important microscopic feature that we look for to distinguish the spores from other things in the stool. So this is the modified Gram chromatrope stain. Histopathology, bottom left, can demonstrate the organisms in the enterocyte cytoplasm, and you can see the arrowheads there pointing to these clumps of organisms. Now, this stain is not H&E, it may be a tissue gram stain or something like that. H&E itself may not show it up very well. Then we have EM, and there's some examples bottom middle. The same species, E. benusi, but in different planes. In cross-section or transverse section, where we can see and count the number of tubules and look at their arrangement. So here you can see they're arranged in 2 rows, which is characteristic of this species. And in this one, a more oblique section, which shows the a bit more of the coils inside the spore. And then we come on to, which is probably the most accurate and most sensitive way of diagnosing and identifying this pathogen, and that is molecular method. And there are a number, and I'll tell you a bit later about some of the molecular methods we use. So now I want to quickly go through some clinical cases, and these were transplant patients with acute graft rejection. And we received samples from 3 patients. And those are the left-hand column shows the dates that we received them. So late November and late December, from Grooterschuur and Red Cross respectively. The first case was a male. They were all renal transplant patients. And we received urine from all 3, and then a bit later, some biopsy sections from the last case. So not to spend too much time on the details here, but basically they were all renal transplant patients, as I said, with suspected or actual acute graft rejection. And I guess not unexpectedly, they had a number of different problems related to the transplant and immunosuppression. But the first case, after going through quite a stormy, well, they all went through stormy illness. Finally, anatomical pathology in Cape Town identified microsporidia in the renal graft, infection in the renal graft, and started the patient on albendazole. The second patient, so patients 2 and 3 were young girls. In fact, they shared the donor. It was a cadaveric transplant, and again also went through acute rejection illness with other opportunistic infections, and again suspected microsporidia, and started on treatment with the urine specimens sent to us. And then patient 3, well, very similar, just a different spectrum of problems, but again, acute graft rejection and suspected microsporidial infection started on albendazole. So I would like to show you what we found in our laboratory. So we were really sent samples to confirm the diagnosis and to see if we could come up with the name of a pathogen here. So this is urine stained with calcifluor white, and this is a nice demonstration because urine, apart from the pathogen, was fairly clear, and so there wasn't too much to interfere with the image. So here you can see these very bright blue, electric blue spores in clumps in the urine. This is another microscopic method called differential interference contrast, or Namaski microscopy, and showing the spores very nicely, some of which, especially in the next one, you can see are empty. So they've ejected their contents, and those are the empty spore coats that you can see in some places. So the urine was spun down and sent to Monica at our EM unit, Monica Burkhead, for electron microscopy, and she was able to readily demonstrate these immature spores still in development in this cell here, probably a sloughed off renal cell. And at higher power, the internal structure, which suggests which species this is, in terms of the number of turns or gyres of the polar filament and the single row. And here's another one, again showing the features of internal features of the spore. So that's a microscopy on urine sediment. Now, if we look at the biopsy samples that we got, this demonstrates another method to show up the pathogen. If you look at the left-hand panel, this is a straightforward H&E, showing renal tissue with some inflammatory infiltrate. But the right-hand panel is with polarized light. Now, these spores have the property of birefringence under polarized light, and so they show up, the mature spores show up as these bright, shiny objects. And under polarized light, you can see very clearly that they're scattered throughout the tissue in this renal sample, renal biopsy sample. And just at higher power, H&E on the left, well, you can't really see much. There's some little sort of articulate things there, but if you look at the polarized light version of the same area, you can very clearly see there's a clump of microsporidial spores in the tissue there. And we can demonstrate the spores as well in the same sample using our other staining methods. So this is the modified gram chromatrope or hot gram chromatrope method, and showing these purplish blue spores in the renal tissue. And this is the modified trichrome stain. So yet another method. And I showed these earlier in terms of stool examination, which may not have convinced you, but I think here you can see that they show up in this sort of sample anyway. They show up the organisms very nicely. So with all these samples, we ran our PCR. Now we've got several PCRs for microsporidial infections, all based on a small subunit ribosomal RNA gene amplification. We have a multiplex for the three common species, E. bernusii, E. cuniculi, and E. intestinalis. Then we have an expanded PCR, which picks up most human pathogenic species. And then we have a universal one, which picks up most microsporidial species. And the last one here was, or the second one, I can't remember now. Anyway, picked up, was PR positive for something. And on sequencing, we showed that it was Encephalitozoon cuniculi, which is one of those important human pathogenic species. And the sequencing showed that the strains from the two little girls were probably the same, and therefore they probably shared a source whereas the strain from case 1, the adult male renal transplant, was different. He, of course, was infected somewhat earlier than the children. So at the time, the question was, is this a nosocomial outbreak of some sort? Well, not necessarily. These are well-known opportunists, and infections in transplant patients are well-known. In fact, the renal unit some time ago at Groote published one of the first renal transplant associated cases. So it doesn't necessarily mean that this is an ongoing risk in that unit. I mean, I don't know what the subsequent history of transplants there might be, whether they've had other cases, but Intracytozoon cuniculi is quite has got quite a wide host range. It infects quite a range of animals as well as humans. In fact, the name comes from infections in rabbits, where it was first described. But interestingly, some dog breeds are susceptible to microsporidiosis due to Intracytozoon cuniculi. I think it's beagles, maybe it's bassets. The disease is well known in this country to fanciers of those dogs, I guess. Now, this outbreak was, the cases in the children were written up in Pediatric Transplantation, 2014, if you want to look at the original article. Now, Monica Birkhead, our EM expert, also published a very nice case study on the various microscopic techniques used to diagnose and identify the disease. Now this was published in a book, not really available, but if anyone wants a copy of the article, just let me know, and I'll send it. I'll send it to you. There's some very nice pictures in this report. Okay, so moving on to another case, a woman with a chronic illness and a muscle mass. Now, the reason why this is somewhat vague is because I couldn't get any cooperation from the clinicians involved in this case. So, but I did have a lot of laboratory reports. So a lot of what I believe the symptoms are extrapolated from the lab in lab report request information. So it seems that she probably started in July 2016 with a diarrhea, because there was a request for stool MCNS and the stool was loose. Syphilis was tested for. A bit later, somewhat later actually, the next year, again, it seems she had diarrhea, and she had endoscopy, which showed basically inflammation throughout the gut. And then a bit later in that year, or substantially later in that year, of course, various other investigations were done, which included TB, CMV, herpes simplex, serology, and syphilis, and autoantibodies. So clearly there was some systemic process going on here. Myoglobin level was requested, so presumably this was the hint that the muscle was involved. And then in October 2017, it looks as though the penny dropped. and she was tested for HIV and found to have a CD4 of 2 with a viral load of 146,000. They did a beta-D glucan, which was non-contributory, and toxo antibodies negative. This proceeded to a muscle biopsy, or a muscle ultrasound, and a mass was found and a biopsy done. At this point, blood cultures were negative, and Cragg was negative on blood. And she demised towards the end of October. And over this period, a number of non-specific laboratory investigations were done. FBCs, LFTs, some activity tests, and also some muscle enzymes. So, not to waste too much time on this, just to summarize all these investigations, this is the timeline of hematology. So HB was sort of at the lower limit of the normal range. That's that orange color here, dropped a bit right at the end. The white cell count, again, tended to be a bit above and a bit below the lower limit of the normal range, went up right at the end slightly. Platelets were generally all right for all the time. And lymphocyte count, there was certainly a lymphopenia tendency right at the end of her illness. Liver functions, well, the enzymes, or some of the enzymes, the ones that were reported were gamma GT, ALT, AST, were moderately raised, went up towards the end. Alk-Phos was only tested once, was a bit above the limit, and albumin was also rather low right at the end. So I think we're seeing the sort of evidence of an ongoing chronic non-specific illness, and they're the activity markers. CRP raised, but not excessively. PCT actually not up much at all. ESR somewhat up around. So these are, sorry, just to explain, this is a log scale at the left, just to accommodate the range of values, and ferritin substantially up at the end. And the muscle enzymes, so the blue line here is the upper limit of normal. So this is the rapid troponin T, so up somewhat. A bit later, the highly sensitive troponin, also that went up, or was up. CK up the most of the time, and the CKMB ISA enzyme also above, high. So, just to summarize all that, here we have this woman at age 33, a chronic illness, which included bowel symptoms and muscle pain. The final event was the emergence of the discovery of a tender mass in the thigh muscle. She was HIV positive, CD4-2, viral load 146,000, enzymes up, muscle enzymes up, and muscle ultrasound showed a mass. They weren't sure what it was, so they biopsied it, and other investigations were all negative. So the histopathologist wasn't sure what she was looking at, so they asked us to help. Basically, there was an inflammatory myositis. They did see organisms which were PAS negative, GROCOT positive. So that's not in keeping with a fungus, because you'd expect PAS positive. So sorry, let me go back. This is muscle fibers in transverse section. So you can see some of them being completely replaced by this massive organisms. Longitudinal section, again, muscle fibers replaced by this mass of organisms. This is a Giemsa stain, sorry, showing them at higher power here. And if I can point out, most of them have got this vacuole in them, and they are grouped together in clumps, separated by walls or septa, which is suggestive of certain Microsporidia. So just on this appearance alone, we could say these are definitely Microsporidia. This is the Grocott. So similar staining properties to yeast, but no evidence of budding, and really not the growth pattern that you would expect to see with yeasts in those septate compartments. Right, so now this is the H&E on the left, and on the right, really the thing which can clinch it microscopically, birefringence under polarized light on the same section. So we know for sure that these are Microsporidia, and confirmed by EM on some of that muscle biopsy material. Now, this was not done by us at NICD. It was the anatomical pathology EM unit at medical school. And our PCR on this patient showed that it was a pathogen called Trachypleistophora hominis, with a large degree of certainty. And this is a well-known, this is a known cause of myositis in immunocompromised people. The myositis, in fact, is not particularly common, but it's well described. And it's the usual risk factors, including people on biologicals for rheumatoid disease and TNF and antagonists. Only one report of microsporial myositis in an immunocompetent patient. Well, it was at that stage. I really haven't looked more recently. And really, at that stage, not a very commonly reported disease. And there are a number of species associated with myositis. And the one of interest to us in this case was Trachypleistophora hominis. And so far, an animal or insect host, natural host for this pathogen hasn't been found, so far only described in humans. So what we require for recognition of microsporidial myositis is a high degree of suspicion in patients who are at risk with muscle weakness, myalgia, muscle swelling, may affect muscles of the face and pharynx. There may be changes on EMG, but definitive diagnosis is really histology and PCR. Therapy, albendazole, which is active against many microsporidia, although not all, plus supplementary antibiotics. So this is the last group of patients, HIV positive individuals with eye problems. So this was the first one, a known HIV positive case, CD4 of 16, who presented with scratchy eyes and blurring of vision. And on fluorescein staining, you can see this punctate keratitis in both eyes. The scraping was sent to us. This is our calcifluor white again, showing up the spores nicely. Deems are on the left, sorry, on the right, and they showed up as these tiny little blue things, very not very specific, and really only seen if one was looking for them. And he was treated with topical fumagillin, which is a standard, a well-known treatment to ophthalmologists and seems to be available in this country, and resolved well, apparently. The other case was more recent, and this is a patient who developed a corneal abscess with erosion of the cornea, and the picture on the left shows what's called a decimatoceal, which a Descemet's membrane is on the back of the cornea, internal surface of the cornea, and when the cornea is damaged, it can herniate through to cause a descemetocele, and that's what's shown there with the surrounding abscess. So a patch was done, and that's the result there, and presumably put on systemic treatment, and ultimately corneal graft was done. From the diagnostic point of view, again, we have the spores showing up on calcifluor white stain, a modified trichrome in the middle at the top, on the right at the top, modified trichrome plus Nomarski microscopy, and then, excuse me, EM at the bottom, done by Monica, and it's probably this organism, Ankylia algeri, which is a well-known cause of keratitis. We weren't able to amplify it and get a PCR result. There simply wasn't enough tissue. Vitiforma corneae is one of the classic causes of microsporidial keratitis, but it definitely wasn't that species. Morphologically, it was not the same. Vitiforma has two nuclei, and the one we had was definitely not. So just to round off, a treatment for microsporidiosis, if the patient's immunocompromised, really it's to restore immune function. And in HIV, of course, put them on ARTs, and basically for microsporidial diarrhea, once the CD4 gets to more than 100, it should resolve. Albendazole, active against many species, but not good for Ebeneuse or Vitiforma, by the way. Various other ones have been suggested. Nitazoxanide, unfortunately not available here. Oral fumagillin, I'm sure not available here, but it's also a toxic drug. For disseminated disease, itraconazole can be added to albendazole for Ankylia and trichoplastophora. Topical fumagillin for keratoconjunctivitis. Probably wise to add albendazole as well, but other topical antifungals and antibiotics have been suggested. If there's stromal keratitis, as in the case I showed you, early keratoplasty is suggested. There are a number of agents which are not active: metronidazole, atovaquone, azithromycin, doxycycline, shouldn't be used for treatment. And I think maybe I should stop there, Jeremy. We had a little outbreak in the insectary with mosquito disease, but I think that's not really relevant to humans, except that some of those mosquito pathogens can infect humans, actually. But these were sick mosquitoes. Well, let me just show you, just for fun. There's a sick mosquito on the left, healthy ones to the right of it, and then the EM showing the pathogen, which is probably, again, an Calia species. But we never got around to doing the definitive identification. And just last point here is they're widespread as insect pathogens, which suggests possible use as biological control method, but there are problems with using natural pathogens for vectors because they basically evolve together, so you'll never adequately control them. But this is a species which was found to infect mosquitoes, not cause illness, but affect the growth of malaria parasites in the vector mosquito. So it suggests a possible way to, if you can get this organism to spread, which may suggest a way to, a method to help control malaria. So I think that really is the end. Thanks.
Thanks, John. That's fascinating and also a little bit scary. I mean, I guess the first question is how do you find out about sick mosquitoes? as in, do they whine more?
Well, they die as larvae. If you look at this, the sick larva on the left, I mean, it fills up, its body basically is replaced by Microsporidia in these horrible yellow masses, and it dies. And if you're breeding mosquitoes for a living like our entomologists do, then it's a great concern. In fact, outbreaks in insectaries are well known, and they have to implement better hygiene methods in their insectary.
Okay, all right, fascinating. I guess always the worry for us, you know, as clinicians who see a lot of HIV positive patients is how much of this we're missing. We must miss a lot of it, given that it's not visible on any of the routine stains. Even if you say parasites and ova, you wouldn't get a stain that would identify microsporidiosis. I mean, is it becoming part of the sort of multiplex stool PCR panels that are commercially available? I know you said you've got your sort of in-house PCRs within the NICD, but I mean, as part of routine diagnostics, at least from a PCR perspective, do you think it'll make it onto those lists soon? Or because
yes, I certainly think it should. If it's not there already, it may well be on some of those commercial multiplex things. Certainly, I mean, clearly you can't cover all the possibilities, but the important ones like Intracytozoon benusi and Encephalotozoon intestinalis belong there for sure.
Yeah, it's a trick, and I see also you said that albendazole wasn't necessarily universally active against some of the more common strains, so that's another problem.
No, it's not. Especially with benusi is not.
Yeah,
it may control it to some extent, but it doesn't eliminate it.
Yeah, because it's not an uncommon scenario. When we see these patients with advanced HIV and chronic diarrhea without any pathogen identified. It's in that fairly enriched sample. I bet a bunch of it's this, but it's very hard to prove. As you say, fortunately, it does get better with immune reconstitution, which is generally our approach when we can't find a pathogen.
Yeah, certainly. I think you should think about asking us to look for it if in that sort of scenario. Yeah.
Sure. And like on a stool sample you took, or would you need a biopsy?
Yes.
I mean,
No, stool
yeah.
sample. Yeah.
Okay, interesting. Yeah.
Because we're gearing up to do PCR basically.
Sure.
The microscopic methods are really not sensitive enough.
Mm. And then for the HIV patients, is it a new infection or can it be a reactivation? I mean, can this exist latently in HIV positive people?
I would think so. If they're, depending on the state of their immune system, I suspect carriage. If you can get carriage in immunocompetence, then for sure you can get carriage in people who are maybe not, ultimately immunocompromised, but still enough, still able to resist the infection to some extent.
Yeah, no, thanks. I see there's a question in the chat about cotrimoxazole. I'm not sure. Sorry, did you mention it was susceptible? I didn't think it was, to be honest.
It's one of the drugs I think added to treatment for systemic infections as a substitute for sulfadiazine.
Okay.
As with the toxo, because we don't have
Sure.
sulfadiazine here. No.
And do we have any sense whether the Bactrim prophylaxis may keep this in check, or do we think it's probably not active enough?
I think probably not active enough.
Okay, interesting. Okay.
Yeah, I haven't seen that as a recommendation. Yeah. Sure.
I think that's it for the questions. That's a fascinating lecture, John, and thank you so much, and opening our eyes, certainly myself included, about some of the possibilities here. And it really does sound like a really fascinating organism as well, and its way of reproduction, and it does seem quite left field, as the Americans would say, for that. But that's a fantastic talk. Thank you so much, John. Really, really helpful.
That's a pleasure. I'll stop sharing now.